Most welcome in the series of science and technology. And see here, so this was the last discussion, right? This was the last discussion, and we have discussed about. Please confirm. I have just shared before the class. I have shared PPTs in the form of PDF to everyone. I have just shared PPTs in the form of PDF to everyone for the today's class. Means what we are going to discuss, the content is already been shared. So please confirm whether you have received this content or not. Already it is uploaded. I was confirmed. My content is already been uploaded. So whether you got this uploaded material or not, please let me know. Confirm. If yes, please let me know. Right. Aditya Chaudhary is confirming yes. So the entire material that you received is for the content of today's class. Right? So please go ahead with the uploaded material. So what we have discussed in the last discussion, in the last lecture, and it was all about right. So it's a PDF of today's class. Means whatever the discussion we are going to discuss, the content is already been uploaded. And only value addition is required. So whenever you feel, I am going to explain everything, even the written part. Already I have uploaded on the application part, so that you feel comfortable and you don't feel like lagging behind in the process of the writing. That's why even before the class I have uploaded this material, right? So just go through the material, and whatever the discussion we are going to discuss, just try to mark it and go for the value addition. Right? So the entire discussion is already been uploaded. Fine. So we have discussed about the genome. Right? And in the previous discussion, we have discussed about the genome, and I told you genome is what? That is the all set of the genetic material of an organism. And for the human, this is 20,000 to 25,000 gene sequences. Clear? And even in the meanwhile, we have also discussed about material number 2, right? Thank you. Sadhana, Meena, everyone has confirmed. Material is already been uploaded. Share, not yeah. It is already been confirmed by everyone except you. The consent is already been streaming on my portal. Fine. So please just try to. So, see here, now this genomics, we have discussed about the genomics, and the genomics is what? It's a study of gene. It's a study of the structure, function, and evolution of the gene. And in this context, in this context of the genomics, we have discussed about plant genomics. And here we are going to discuss about animal genomics, especially human genomics. Right? So here we are, this part already we have done. It is already been done in the previous discussion, and this we are going to elaborate. So here this concept is what? It's a human genomics, right? So listen, when we talk about this human genome, it means this human genomics So for the human genomics, this human genome project was first of all launched in 1990 and this project was terminated in 2003. So this human genome project was launched in 1990 and it was terminated in 2003. So content is given in the booklet. The content is already I have shared. Fine. So no need to write, rather just mark it. Alright? So ultimately the goal of this human genome project was just to identify 20,000 to 25,000 gene sequences of the human, and these 25 to 25,000 gene sequences are made up of 3 billion base pairs. 3 billion base pairs means this statement is trying to make you understand like this is human chromosome that consists of 23 pairs of chromosome that itself consists of 20,000 to 25,000 gene sequences, and this itself is made up of 3 billion base pairs in DNA. 3 billion base pairs in DNA. Fine. Then we need to store this information. We are exactly in the database. So ultimately, the intention of the human genome project was just to identify 20,000 to 25,000 gene sequences, determining the sequences of the 3 billion chemical base pairs, and these base pairs are nothing but these base pairs are the 80 GC base pairs. I told you in the last lecture, DNA has 80 and the GC base pairs, and then further storage of these information in database. Storage of these information in database. So after storage of these informations in the database, from here onward, a new branch was initiated, and this branch is what? It's a bioinformatics. It's a bioinformatics, means the branch of information technology that consists of, which is involved in the storage of the biological data that make a sense of bioinformatics. The information technology which involves storage of the biological information is the bioinformatics, right? Bioinformatics. Fine. Okay. So that was the ultimate goal of human genome project that was started in 1990 and then officially terminated in 2003, right? Fine. Again, see the silent feature of this human genome means finding. We are talking about the finding. The human genome consists of approximately means 20,000 to 25,000 gene sequences. Fine. That is the first point. The second point that we need to consider like this. Almost 99% of nucleotide bases, means 99% of the gene sequences are exactly same in all people. Means it is saying that me and you means across the world 99, right? All humans are similar in context, means more than 99% of the gene similarity we are showing. More than 99% of the gene similarity we are showing. That is the finding of human genome project. Not 100%. It's a more than 99% of the gene similarity between the two individuals. Fine. Okay. Not 100%. It's a more than 99%. Chromosome 1 has most of the gene sequences, approximately 3,000, while the Y has the lowest gene sequences. This is the minimum, like 231 gene sequences. Means the number is not required, what we are supposed to consider here, the chromosome number 1 has maximum gene sequences that already I have discussed earlier, while the Y chromosome has the minimum gene sequences. Minimum gene sequences. Clear? So this is the finding of Human Genome Project. It's the finding of the Human Genome Project. And ultimate application of this Human Genome Project is what? Once the human genome is sequenced, we could go for easy determination of genetic sequences, and in the case of any gene defect we are going to observe, right? So we could be able to identify it quickly in a very easy manner, just because the sequences are already been sequenced under the Human Genome Project, right? Fine. So see here, if any disease, if any gene is faulty, we should go for gene correction. So gene correction, that is the gene therapy, became possible after genome sequencing. Gene correction is possible after genome sequencing. Gene correction is now become possible after genome sequencing. And nowadays we are just trying to handle so many gene defects, right? The success rate is even less than 1%, but even we are trying to find out the correction of genetic fault through the gene therapy. What exactly we are doing, that we are going to elaborate. So the point is that the application of this Human Genome Project is what? That is finding the way to cure gene therapy, and not only gene therapy. You know, if the gene is sequenced, we should also try to find out the evolutionary pattern among the different races of the human. Like you heard about a different human species like the Cro-Magnon Mano, Cro-Magnon human, right? Neanderthal, fine, Homo sapien erectus, Homo sapien sapien, fine. So ultimately we are just trying to find out the evolutionary pattern, and that could be possible only after sequencing of the gene sequences, right? Sequencing of the gene sequences, fine. Okay, so that was the first human genome sequencing project that was started in 1990, and then it was officially terminated in 2003. Clear? And then after that, this entire discussion, the entire effort opened door for the next level of the genome sequencing that was started independently by so many countries. Like see here, the Siddhant, we are going to discuss about the CRISPR-Cas9 after half an hour. So just wait for. We just started the human genome sequencing, and this CRISPR-Cas9 will come somewhere in the gene therapy. Fine. So just hold your question. We are going to discuss probably after half an hour this discussion, right? So when we talk about this genome sequencing, this UK, United Kingdom, UK has launched Genomics England. This is a project. Fine. Australia has launched 100,000 genome projects, and the intention is what? Sequencing the patient with the rare disease and cancer to create the massive database and the research and development. Clear? The ultimate purpose of the sequencing of the genome is what? Suppose if there's any problem, suppose if you are going to sequence gene, so will be, and even we succeeded, so we are going to discuss, we are going to find out the entire discussion about genetic defect, or even the rare diseases like the cancer, or even so many metabolic disorders we are going to correct after observing the genome sequences, right? Again, you see here. USA started Precision Medicine Initiative independently. It's a human genome project. In 2016, French announced France Medicine Genomic 2025 project. Then again, non-profit consortium Genome Asia 100,000 decided to generate genomic data for the Asiatic population, supported to include genomic companies like ultimately the Genome Asia started 10,000. Genome Asia started 100,000 projects. So these are the independent projects which was started by independent individual countries, like the UK started this, Australia started this, US started this, France started this, and the independent non-NGO consortium started, Genome Asia started 100,000 project. And ultimately the intention of the entire project is what? That is the sequencing of the genome. Fine, sequencing of the genome. And the sequencing of the genome is what? Sequencing of the gene, and after the sequencing of the gene, we'll try to find out the structure of the gene. And if the sequencing of the gene is done, and if the structure of the gene is known, we should try to, we'll be able to justify the function of the gene, right? Then this project was also started independently in India. And this project is what? This is the Indian project. And this Indian project it's an indigene project. It's an indigene project, and this project was started by CSIR. And the intention of this project is to understand the extent of genetic variation in Indians. And here CSIR drafted 1,000